diving into bioinformatics today. Got a whole stack of stuff about genes and genomes and all that. Looks like we're going to be figuring out how computers help us make sense of all this biological data, huh?
Yeah, it's like having a super powerful magnifying glass to look at all the little details of life. Bioinformatics can help us answer all sorts of questions, questions that we couldn't even think about answering before, like how genes actually work, or how organisms evolve, or even how to create personalized medicine.
Right, yeah, and I'm seeing stuff here about, like, identifying genes that cause diseases, and even understanding how whole species evolve. Pretty amazing stuff.
Yeah.
One of the things that really pops out to me is these bioinformatic databases. They seem to be like the foundation of this whole field.
Oh yeah, absolutely. You can think of them like giant digital libraries, but instead of books, they have just tons and tons of information about genes and proteins, diseases, you name it. Without these databases, we'd be totally lost, like trying to find a needle in a haystack.
Right, that makes sense. But I'm looking at one of the sources here, and it talks about an early database that used this flat file format. And they have this example here. It says Nancy, pipe, Dengler, pipe, botany, pipe, and it's a mess.
Oh, it was a mess. You can imagine trying to find a specific book in a library where all the books are just piled up randomly. That's basically what it was like with those early databases. No real structure, made it super hard to find the information you needed.
So how did they fix that?
Well, that's when relational databases came along. They organized everything into these nice, neat tables with links between them, sort of like a library card catalog. Made it way easier for researchers to search for specific things, like the name of a gene or what it does or even what diseases it's related to.
Makes sense. That definitely sounds more organized. But there's still so much information in these databases, and I'm seeing all these weird codes in the material, like KY5494441.1. What are all these codes about?
Oh, those are accession codes. Each entry in a database gets its own unique code, like a fingerprint. That way you know you're looking at the right information no matter what database you're using.
So if I'm researching a gene, let's say, I can use that code to find it in any database.
Exactly. It's like a universal language for genetic information.
Yeah.
And it helps to make sure that everyone's on the same page.
Okay, that makes sense. But here's another thing that's a little overwhelming. There are so many different databases out there. The material mentions GenBank, UniPeyote, Flybase, the Human Protein Atlas. It's hard to keep them all straight.
Don't worry, you don't need to be an expert on every single database. It's more important to understand that each one has its own strengths and focuses, what it's best for. Like take GenBank, for example. That's like the main library for DNA and RNA sequences, just a massive collection of genetic information.
So if I wanted to find the DNA sequence for a specific gene, GenBank would be the place to go.
Yep, exactly. And then there's UniPro-ROTE, which is all about proteins. It's like a giant encyclopedia of protein information. It tells you about their structure, their function, how they interact with other proteins.
What about those other ones, Flybase and the Human Protein Atlas? They sound pretty interesting.
Well, Flybase is all about the fruit fly, which has been used in genetic research for ages. It's a really important resource for understanding those basic biological processes, like how genes work.
And I've heard that fruit flies are actually surprisingly complex.
Oh yeah, definitely. And then there's the Human Protein Atlas, which is like a map of where different proteins are found in the human body. It really helps us understand human biology and disease on a molecular level.
Wow, that's fascinating. Speaking of maps, the material mentioned these terms, orthologs and paralogs. Honestly, they sound kind of intimidating.
They're not too bad once you get the hang of them. Think of orthologs as genes in different species that share a common ancestor. Like, for instance, the gene that makes hemoglobin in humans, there's a similar gene in mice, even in fish.
So, like, they're distant relatives in the gene family.
That's a great way to think about it. Now, paralogs, those are genes within the same species that came about through duplication. It's like a gene got copied, and then each copy evolved on its own.
So orthologs are like cousins, and paralogs are like siblings. But why is it so important to understand all this gene stuff anyway?
Well, it helps us understand how genes work and how they've changed over time. For example, you can compare orthologs in different species to figure out which parts of a gene are really essential. Those are the parts that tend to stay the same over millions of years because they're so important.
Like finding the building blocks that are common to all life.
Exactly. And then paralogs can show us how genes evolve new functions. One copy might keep the original function while the other one mutates and, maybe takes on a whole new role. It's how life gets more diverse and complex.
That makes sense. Like evolution is experimenting, seeing what works. But how do researchers actually use all of this knowledge? I mean, how do they actually use these databases to answer real-world questions?
That's where things get really exciting. Let's take a look at this example from the material, The Search for SNPs related to early onset breast cancer.
Yeah, that one caught my attention too. It looked pretty complicated. Where do you even begin with something like that?
Well, they started with a database called OMIM, which is all about human genetic diseases. It's like a specialized library that helps us understand the genetic causes of different conditions.
So they used OMIM to learn about the genetics of early onset breast cancer?
Right. And then they use the NCBI database, which is like a central hub that connects a bunch of different bioinformatics resources. From there, they could find links to other databases with information about SNPs. Those are those single nucleotide polymorphisms that can sometimes increase the risk of disease.
So it's like they're following a trail of clues through the world of genomic data.
That's a great analogy. And those clues, the SNPs, can lead to some really important discoveries. Imagine developing a test that could screen for those SNPs and identify people who are at a higher risk. That's the power of bioinformatics, taking raw data and turning it into knowledge that can actually help people.
That's amazing. It's like bioinformatics is changing the whole landscape of healthcare. Speaking of changing landscapes, the source material mentions this thing called the bioinformatic harvester. Sounds pretty sci-fi.
It's a pretty cool name. It basically automates the process of searching for and downloading data from different databases. Before, researchers had to do all of that manually, which was incredibly time-consuming.
Yeah, I can imagine, like trying to put together a puzzle where all the pieces are scattered everywhere.
Exactly. But with the bioinformatic harvester, it's all done automatically. It's like having a research assistant that gathers all the data you need from all over the place and puts it in one neat package.
Wow, that's got to be super helpful. Frees up researchers to do, the more creative parts of their work.
That's the idea. Streamline things, make research more efficient, and it's just one example of how technology is pushing the boundaries of what's possible in bioinformatics.
Technology is really changing the game everywhere, isn't it? What other tech advances are shaping the future of bioinformatics?
Oh, definitely. One of the most exciting areas is the use of artificial intelligence, or AI, and machine learning. These are changing how we analyze and make sense of biological data, opening up whole new avenues of research.
Those are some pretty big buzzwords. But how are they actually used in bioinformatics? It all sounds kind of futuristic.
It does sound like sci-fi, but it's becoming reality. Like I said before, the amount of data we have in biology is just enormous. No human could possibly analyze it all.
Yeah, I'd probably just give up and take a nap.
I get it. That's where AI and machine learning come in. These algorithms can analyze huge amounts of data, identify patterns, and even make predictions in ways that we just can't.
So it's like having a supercomputer that can do the heavy lifting for us.
Exactly. In bioinformatics, they're using AI and machine learning for things like predicting the structure of proteins, identifying new drug targets, even developing personalized cancer treatments. It's a whole new level of precision and insight.
That's incredible. But with all this talk about machines and algorithms, it makes you wonder about the role of humans in research. Are scientists going to be replaced by machines?
It's a question a lot of people are asking, and for good reason. But the way I see it, AI and machine learning are tools. They can help us do things better and faster, but they can't replace human intelligence.
It's more of a collaboration.
Exactly. We can use these tools to process information more efficiently, identify patterns we might have missed, maybe even generate new hypotheses. But at the end of the day, it's still up to human researchers to interpret the results, design experiments, and make those big decisions.
So it's like a partnership between human and artificial intelligence.
Exactly. Combining the strengths of both. And it really highlights how important it is to train the next generation of scientists to be skilled in both computation and biology.
So it's not just about being a computer whiz, it's about being a well-rounded scientist.
Exactly. That's what makes bioinformatics so exciting. It's where cutting-edge technology meets the mysteries of life, constantly pushing the boundaries of what we know about ourselves and the world around us.
I gotta say, I'm even more fascinated by bioinformatics now than I was before. But I'm also a bit overwhelmed by all the different databases and tools and techniques and everything. It's a lot to take in.
I get it. It's a lot. But the key is to start with a question, something you're curious about. What biological mystery do you want to solve?
One thing that really caught my attention was the idea of personalized medicine. The fact that we can tailor treatments to an individual's genes, That's just amazing.
That's a fantastic area to explore. It's a perfect example of how bioinformatics is changing healthcare. There are companies out there already that offer genetic testing to figure out your risk for certain diseases or how you might respond to different medications. It's about moving away from that one-size-fits-all approach to healthcare.
That's incredible. So if I wanted to learn more about personalized medicine, where would I even begin? What databases or tools should I check out?
A great starting point would be a database called ClinVar. It focuses on human genetic variations and what they mean for health. It can help you understand how those little differences in our genes can impact our health and how we respond to treatments.
ClinVar. Okay, I'm going to write that down. And what about tools for actually analyzing genetic data? The material mentioned something about a genome browser.
Oh yeah, genome browsers are essential. They let you visualize and analyze genomic data. You can zoom in and out of different regions of a genome, explore genes and their annotations, even compare genomes from different people or different species. It's like Google Maps for DNA.
That sounds incredibly useful, like having a window into the blueprint of life.
Exactly. There are lots of different genome browsers out there, each with its own strengths and features. Two popular ones that are used a lot in research are the UCSC Genome Browser and Ensemble.
Okay, I'm adding those to my list. I'm starting to feel a little less overwhelmed and more excited about all this.
That's the spirit. Bioinformatics is all about discovery. Just be curious, ask questions, and never stop learning.
I'm definitely feeling inspired. But before we move on, I have one more question. We talked about the bioinformatic harvester for, collecting data automatically. Are there other tools that can help researchers analyze and interpret all that data once they have it?
Of course. That's really where the magic of bioinformatics happens, turning all that raw data into something meaningful. And there's a whole bunch of tools out there, each one designed for a specific purpose.
Can you give me some examples? I'm curious what's out there.
Sure. For example, there are tools for sequence alignment, which let you compare DNA or protein sequences to find similarities and differences. That's super important for understanding evolutionary relationships, seeing which parts of a gene are really important and even predicting what a protein might do.
So it's like finding the common threads in all that genetic code.
Exactly. And there are tools for phylogenetic analysis, which helps us figure out the evolutionary history of different organisms based on their genes. It's like building a family tree for all life on Earth.
It's amazing how we can trace all those evolutionary connections back through time.
It really is. And then there are tools for gene expression analysis, protein structure prediction, pathway analysis. The list goes on and on. It's a constantly evolving field with new tools and techniques being developed all the time.
It's incredible to think about all the possibilities. Like, we have this amazing toolkit to help us understand life at its most fundamental level.
And the best part is that this toolkit is becoming more and more accessible to everyone with open source software, cloud computing, online tutorials. Anyone with an internet connection can start exploring the world of bioinformatics.
That's fantastic. It's like we're democratizing access to all this knowledge, making it available to anyone who's curious.
Exactly. And that's what makes bioinformatics so exciting. It's driven by collaboration, innovation, and a shared passion for understanding the wonders of life.
I'm feeling pretty inspired after this deep dive, already thinking about all the databases and tools I want to explore.
That's great. Just remember, stay curious, keep asking questions, and never stop learning. There's always something new to discover in bioinformatics. We were just talking about GenBank, and I think it's worth looking a little closer at how these databases are actually structured. Remember that example from the material, the entry for the ribulose 1-villi-5 bisphosphate carboxylase oxygenase, BISCO, large subunit gene from a geranium.
Yeah, I remember that one. It wasn't just the DNA sequence, though, was it? There was a lot of other information attached to it.
Right, exactly. Like the gene's accession number, what organism it came from, how long the sequence was, even a protein ID. These databases are designed not just to store the data, but to give it context, make it useful for researchers.
So it's a detailed record for that gene, keeps all the important information in one place.
Exactly. And that's where those accession codes we were talking about come in. Remember, that geranium gene had the code KY54944.1.1. So anyone who's studying that gene can find that exact entry in GenBank even years from now, makes the data reliable and accessible.
Okay, that makes sense. It's like a universal language for genetic information. But let's go back to those orthologs and paralogs for a second. You mentioned that comparing orthologs can help us figure out what parts of a gene are essential. How does that work?
Well, think about it. If a particular section of a gene is almost identical across, say, humans and mice and even fruit flies, that suggests that section is pretty darn important for that gene to function properly, right? It's been conserved through evolution because it's that crucial.
Oh, okay, so it's like finding the core building blocks that have been preserved over millions of years.
Exactly. And then studying paralogs, that can show us how new functions evolve. After a gene is duplicated, one copy might stick with the original function, while the other one is free to mutate and potentially take on a new role entirely.
It's fascinating how these little changes, these tiny little changes in genes over millions of years, can lead to all the amazing diversity that we see in nature.
It really is. It shows how powerful evolution is. And with bioinformatics, we have the tools to trace those evolutionary journeys, to understand how genes have changed and adapted over time.
So we've talked about databases, accession codes, even how genes evolve.
Yeah.
But I'm still curious about how all of this comes together in, real research. How do scientists actually use these tools to answer questions?
Well, remember that example we touched on earlier, the search for SNPs related to early onset breast cancer. That's a perfect example. Shows how researchers can navigate this whole network of databases to piece together a complete picture.
Right. They started with OMIM to understand the genetic basis of the disease. Then they used NCBI to find those databases with information about SNPs. It's like detective work, but on a molecular level.
Yeah, it's a great way to think about it. And those clues, those SNPs, can lead to big breakthroughs. Imagine being able to develop a genetic test that can screen for these SNPs and identify people who are at higher risk of developing early onset breast cancer. That's the kind of impact bioinformatics can have, taking data and turning it into knowledge that can potentially save lives.
It's incredible. It really is changing healthcare. Speaking of changing things, the material mentions something called the bioinformatic harvester. It sounds like something straight out of a sci-fi movie.
It's a pretty fitting name when you think about it. Traditionally, researchers had to manually search and download data from all these different databases. Really time-consuming process.
Yeah, I can imagine that getting pretty tedious, like trying to put together a huge puzzle with the pieces scattered everywhere.
Exactly. But the bioinformatic harvester, it automates that whole process. It's like having a personal assistant who gathers all the relevant data from multiple sources and gives it to you in a nice, organized package.
That sounds amazing. It would free up researchers to focus on the bigger picture instead of getting bogged down in all that data management.
Exactly. It's all about streamlining the process, helping scientists be more efficient. And it's a great example of how technology is constantly pushing the limits in bioinformatics.
Technology is changing the game in so many fields these days. What other kinds of technological advances are shaping the future of bioinformatics?
Well, one of the most exciting areas right now is the integration of artificial intelligence, machine learning, and big data analytics. These technologies are changing how we analyze and interpret biological data, opening up entirely new avenues of research.
Those are some big buzzwords. But how are they actually being applied in bioinformatics? I'm curious because it all sounds a bit futuristic to me.
I know what you mean. It might sound like science fiction, but it's quickly becoming reality. Like I was saying earlier, you have these data sets with millions, even billions of data points. There's no way a human can sift through all that information and make sense of it.
I think I'd just throw in the towel and take a nap.
I hear you. But that's where AI and machine learning come in. These algorithms can analyze massive data sets, identify patterns that we might miss, and even make predictions that would be impossible for humans to do on their own.
So it's like having a supercomputer that can do all the heavy lifting for us.
Yeah, that's a good way to put it. And in bioinformatics, they're being used for everything from predicting the structures of proteins to identifying potential drug targets to developing personalized cancer treatments. It really is a whole new level of precision and insight.
Wow. That's incredible. But with all this talk about machines and algorithms, I can't help but wonder about the role of human researchers. Are they being replaced?
That's a question a lot of people are asking these days. But honestly, I think that AI and machine learning are tools that can actually enhance human capabilities, not replace them. They can help us to process information faster, see patterns that we might have missed, and generate new hypotheses. But ultimately, it's still up to human researchers to interpret the results, design experiments, and make decisions based on their expertise and intuition.
So it's more about collaboration than competition, like a partnership between human intelligence and artificial intelligence.
Exactly. It's about leveraging the strengths of both to advance our understanding of life. And it highlights how important it is to train a new generation of bioinformaticians who are skilled not only in computational techniques, but also have a deep understanding of biology and scientific method.
So it's not just about being a computer whiz, it's about being a well-rounded scientist who can bridge the gap between these two worlds.
Exactly. And that's what makes bioinformatics such a dynamic and exciting field right now. It's where cutting-edge technology meets those mysteries of life, and it's constantly pushing the boundaries of what we know about ourselves and the world around us.
I have to say, I'm even more fascinated by bioinformatics after hearing all of this. But I also have to admit, I feel a little overwhelmed by all the different databases, tools, and techniques that we've talked about. It's like drinking from a fire hose.
I completely understand. It is a vast and complex field, and it can be hard to know where to even begin. But remember, the key is to start with a specific question or problem that you're interested in. What are you curious about? What biological puzzle are you trying to solve?
One of the things that really grabbed my attention was the concept of personalized medicine. The idea that we can tailor treatments to an individual's unique genetic makeup is just incredible.
That's a fantastic area to explore. It really is a perfect example of how bioinformatics is driving innovation in healthcare. There are already companies that offer genetic testing to identify an individual's risk for certain diseases and predict how they might respond to different medications. It's about moving away from that one-size-fits-all approach to medicine and toward a more personalized and effective form of healthcare.
Wow, that's just amazing. So, if I wanted to learn more about personalized medicine, where would I even start? What databases or tools should I check out?
Well, a great starting point would be to explore databases like ClinVar, which focuses on human genetic variations and their clinical significance. It can help you understand how specific genetic differences can affect an individual's health and how they might respond to certain treatments.
ClinVar. Okay, I'm making a note of that. And what about tools for actually analyzing genetic data? The source material mentions something about a genome browser.
Ah, yes, genome browsers. Those are essential tools for visualizing and analyzing genomic data. They allow you to zoom in and out of different regions of a genome, explore genes and their annotations, and even compare genomes from different individuals or species. It's like having Google Maps, but for DNA.
Wow, that sounds incredibly useful. It's like having a window into the blueprint of life.
Exactly. And there are many different genome browsers available, each with its own strengths and features. Two popular options that are widely used in the research community are the UCSC Genome Browser and Ensemble.
Okay, I'm adding those to my list. I'm starting to feel less overwhelmed and more like I'm on an exciting adventure.
That's the spirit. Remember, bioinformatics is a journey of discovery. Embrace the complexity, be curious, and don't be afraid to ask questions.
I'm definitely feeling inspired. But before we move on, I have one more question. We talked about the bioinformatic harvester as a tool for automating data collection. Are there other tools that can help researchers analyze and interpret this massive amount of data once they have it?
Absolutely. That's where the real power of bioinformatics comes in, turning that raw data into meaningful insights. And there is a whole suite of tools available, each designed for a specific purpose.
Oh, wow. Can you give me some examples? I'm really curious to know what's out there.
Sure. For starters, there are tools for sequence alignment, which allow you to compare DNA or protein sequences to identify similarities and differences. This is really important for understanding evolutionary relationships, identifying conserved regions, and even predicting protein function.
So it's like finding those common threads in the tapestry of genetic code.
Exactly. And there are tools for phylogenetic analysis, which help us reconstruct the evolutionary history of organisms based on their genetic relationships. It's like drawing a family tree for all life on Earth.
Wow. It's amazing how we can trace those evolutionary lineages back through time.
I know, right? And then there are tools for gene expression analysis, protein structure prediction, pathway analysis, the list goes on and on. It's a constantly evolving landscape of tools and techniques, each designed to unlock a different aspect of biological complexity.
It's mind-boggling to think about all the possibilities. It's like we have this incredible toolkit that can help us understand life at its most fundamental level.
I agree. And the best part is that this toolkit is becoming increasingly accessible to everyone. With the rise of open source software, cloud computing, and online tutorials, anyone with an internet connection can now delve into the world of bioinformatics.
That's really empowering. It's like democratizing access to this knowledge and making it available to anyone who's curious enough to explore it.
Exactly. And that's what makes bioinformatics so exciting. It's a field that's driven by collaboration, innovation, and a shared passion for understanding the wonders of life.
I have to say, I'm feeling pretty inspired after this deep dive. I'm already thinking about all the different databases and tools that I want to explore.
That's great to hear. Remember, the most important thing is to stay curious, ask questions, and never stop learning. There's always something new to discover in bioinformatics.
We've covered a lot of ground. Databases, genes, even AI in bioinformatics. What's next for this field? What are the researchers excited about these days?
Well, bioinformatics is always changing. There's always something new and exciting happening. But I'd say some of the hottest topics right now are, well, you've heard them before, artificial intelligence, machine learning, and big data analytics.
Yeah, those are definitely buzzwords you hear everywhere. But what makes them so important for bioinformatics?
It all comes down to the sheer amount of data. We're talking about huge data sets, billions and billions of data points. It's just impossible for humans to analyze all of that manually.
I can only imagine. Trying to make sense of all that data would probably make my head spin.
Right. That's where AI and machine learning come in. These algorithms can handle these massive data sets, find patterns that we might miss, even make predictions based on the data.
So it's like having a super smart research assistant to help us out.
That's a good way to put it. In bioinformatics, AI and machine learning are being used for all sorts of things, from predicting how proteins fold, to finding new drug targets, to, developing personalized cancer treatments.
That's incredible. But with all this automation and AI, what does that mean for the scientists themselves? Are they being replaced by machines?
That's a good question, and a lot of people are wondering about that. But honestly, I see AI and machine learning as tools that can help us do our jobs better, not replace us altogether.
This is more about working together, humans and machines.
Exactly. They can help us process information faster, find hidden patterns, come up with new ideas. But it's still up to us, the researchers, to make sense of it all, to design the experiments and use our expertise to make decisions.
It sounds like a powerful partnership. But all of this seems so complex. How can anyone even begin to learn about bioinformatics? It seems a little intimidating.
It can seem that way at first, but there are so many resources available now, more than ever before. Online courses, tutorials, open source software, a lot of it is even free.
That's good to know. So you don't need a fancy degree to start learning about this stuff.
Not at all. The most important thing is to be curious and willing to learn. Obviously, a good foundation in biology and computer science helps, but there are many ways to get into bioinformatics.
Well, this deep dive into the world of bioinformatics has been truly eye-opening. We've explored so much, from the basics of databases and gene evolution to the cutting edge of AI and personalized medicine.
It's been a great conversation, and I'm glad we could share this journey with you.
I feel like we've only just scratched the surface of what's possible with bioinformatics.
You're absolutely right. This field is constantly evolving, and who knows what amazing discoveries are waiting for us in the future.
To all our listeners, thank you for joining us on this deep dive into the world of bioinformatics. We hope you've enjoyed it and are inspired to learn more about this incredible field.
And until next time, keep asking questions and never stop.